Welcome students. We are going to discuss about cell biology. The first topic that we had taken was virus from general biology. The second topic that we are taking up cell, that is the fundamental unit of life. Now before study an organism, the basic cells, then tissues, then organ system and then organelle. This is the organization. If we consider things, the first thing is your cell, then tissue, then organ, then organ system, and then organism. So the fundamental unit of life is cell. All the multicellular organisms are made up of, finally, cells. Similar cell form tissue. Group of tissues form an organ. A group of organ forms an organ system. And different organ system together forms the organism. So the basic structure starts from the cell. The basic structure starts from the cell. Now, moving on to the aspects of cell. The literal meaning of the term cell is a little room or a compartment. So Robert Hooke first observed these boxes or compartment in cork. Cork you get from the bark of tree. Especially in the Mediterranean area, you get cork oak. So Robert Hooke observed the cell in the corks. Based on the cell, there came a theory called cell theory. The theory called cell theory. Cell theory. This cell theory states that all plants and animals, they are composed of cells, and cell is the basic unit of life. This was given by two biologists, Sleeden and Swan. Two biologists, Sleeden and Swan, gave this thing called cell theory. Another thing was added into it was Virchow. He added that cell arises from pre-existing cell. All cells arise from pre-existing cell. The key point is your pre-existing cells. So two main point are first is all the organisms are made up of cells, that is the basic unit of life. Cell is the basic unit of life. And second component is your cells arise from pre-existing. These are two basic. Now the study of cell biology expanded with the expansion in microscopes. The beginning microscope were crude, so we could not see very small things. the beginning microscope, we were able to observe the major structure inside the cell wall and the thing. And then came electron microscope. Electron microscope enabled the scientist to see things at very small scale. Electron microscope can help you see things at angstrom level. Angstrom level. Angstrom is 10 to the power minus 10 meter. So now we are able to see very small things. Other things that were utilized to study the very smaller component and even molecular structure in detail is X-ray crystallography. X-ray crystallography. So in X-ray crystallography, you produce a pure crystal of the substance, say DNA or ribosome, anything you study. Then you put X-ray on it, and this is recorded on a photographic plate in the forms of dot. And then you use mathematical equation to find out the arrangement of atoms. To find out the arrangement of atoms. So X-ray crystallography is one of the most accepted technique in science. You can study the molecular structure, say diamond, graphite, coal, all are made up of carbon, but the difference is in the arrangement of the carbon atom. So using this technique, X-ray crystallography, you can study things at molecular scale. You can study things at molecular scale and also study the arrangement of atoms in the crystal, whether it is DNA crystal or a protein crystal, etc. And in the recent times, there has been a use of a new technique called cryo-electron microscopy. New technique called cryo-electron microscopy. Cryo means low temperature. Cryo means low temperature. So in cryo-electron microscopy, using liquid nitrogen, the molecules are freezed or the cellular components are freezed, and then they are observed under electron microscope. So you use two things. One is liquid nitrogen and electron microscope. So they were used to study many of the virus and other things. That is why 2017 Nobel Prize Chemistry is for cryo-electron microscopy. Sometime UPSC does ask about the recent Nobel Prize. Now moving on to the shape and size of the cell. Shape and size of the cell is related to the function performed by them. Depending on the function, the shape and size of the cell would be variable. For example, the cells in the digestive system, they have some other function. The cells of the excretory system, for example, kidney, have some other function. But there are some cells, for example, there is an organism called amoeba. Amoeba is a single-celled organism. So this is a single cell. Amoeba has the unique capacity to change its shape. Whereas in normal cells, they have a fixed shape. They have a fixed shape. Whereas in case of amoeba, amoeba keeps on changing its shape according to the condition. Even inside the cell, you will find division of labor. Now, Adam Smith economic theory is based on division of labor. Specialization. So, inside the cell also, different components are specialized to perform different function. So, different component performing different functions. Due to specialization, the different cell organelles perform their function, and this helps the cell in doing its function or maintaining its function in an effective manner. So that is why the cell organelles, the organelles inside each organelle perform a special function. So due to this division of labor and specialization, they are able to perform in a better way. Now this is the diagram of a cell. This is the diagram of a animal cell. This one is an animal cell. If we take a look at it, the outermost layer is plasma membrane. The outermost layer is plasma membrane. Inside the cell you have different cell organelles. For example, Golgi body, endoplasmic reticulum, smooth endoplasmic reticulum. Then you have rough endoplasmic reticulum. We use the term RER, rough endoplasmic reticulum, and for smooth endoplasmic reticulum we use the term SER. So you have smooth endoplasmic reticulum and rough endoplasmic reticulum. Then mitochondria, or the powerhouse of the cell, ribosome, the protein factory, lysosome, centriole found near the nucleus, and here this one is the nucleus containing the genetic material over here. So this is the structure of an animal cell. Now moving on to plant cell, first let us take a look at the diagram, then we will see the individual organelle in detail. Here again, one unique feature is your cell wall. Mitochondria and ribosome were common to both plant and animal cell. Then Golgi was there in plant cell also. Here plasma membrane lies inside the cell wall, and this one is vacuole. This center portion is vacuole. This is the nucleus. This one is chloroplast, smooth endoplasmic reticulum, rough endoplasmic reticulum, then lysosomes. These are the structure inside. Then in plant, one specific feature is your chloroplast, which is not there in animal cell. Now let us begin with plasma membrane or cell membrane. Plasma membrane or cell membrane. Let us begin from here. Plasma membrane or cell membrane. This is the outermost covering of the cell. Plasma membrane is what? Outermost covering of the cell. And this separates the primary function of plasma membrane is to maintain the internal environment of the cell. So outside the cell the condition is something else, inside the cell condition is something else. So this helps in maintaining the internal environment of the cell. This plasma membrane is semi-permeable, and therefore plasma membrane allows or allows the entry and exit of some material in and out of the cell. So it is semi-permeable. We use the term plasma membrane is semi-permeable. It allows something to come inside and it blocks other. Semi-permeable. That is why it is also called that the cell membrane or the plasma membrane is a selectively permeable membrane. It means it allows, say, X can move inside and Y is blocked. So X is allowed whereas the Y is blocked. So that is why it is selectively permeable. Selectively permeable. Plasma membrane is made up of two things. Plasma membrane is made up of two things, lipid and protein. This is made up of lipid and protein. One of the unique features of plasma membrane is presence of receptors. Receptors. Many of the hormones and biological molecules get attached to the receptor and then gain an entry inside the cell. Receptors are important. Many of the biomolecules and even viruses get attached to the receptor. You might have heard about a receptor called ACE2 receptor. ACE2 receptor. The COVID virus gets attached to the ACE2 receptor, which are present in the respiratory system, respiratory tract and in the oral cavity, and that is how the COVID virus gains an entry inside the cell. So we have discussed that plasma membrane is selectively permeable. Some substance, like especially the gases like CO2 and O2, they can move across the plasma membrane by a simple process called diffusion. Same is with the case of water. Water also follows the simple process of diffusion across the plasma membrane. Then another phenomenon is osmosis. One is by simple diffusion, water, gases, they move across by simple process called diffusion. Another process is osmosis. Osmosis. Now, osmosis depends on the amount of substance that is dissolved in water. So it is passage of water from a region of high water concentration through a selectively permeable membrane to a region of low water concentration. For example, outside water is more, inside water is less. So what happens? This selectively permeable cell membrane allows the movement of water from outside to inside. Most of the unicellular organisms and plant cell, they gain water by the process of osmosis. The roots and other, they gain water by the process of osmosis. Same in case of your roots. The root cell absorb water by osmosis. The flexibility, particularly in case of amoeba, if you remember, we had discussed while we were discussing this topic, we discussed that amoeba can change its shape. So the cell membrane or the plasma membrane is flexible. So the flexibility allows the amoeba to engulf in food and other material from its external environment. And this process is called endocytosis. This is called endocytosis. And amoeba acquires its food through such process. Amoeba acquires the food through such process. Outside the plasma membrane, there is another structure called cell wall. Outside the plasma membrane, you have another structure called cell wall. So this is the rigid outer covering. Cell wall is rigid outer covering. It lies outside the plasma membrane. Outside the plasma membrane. And cell plasma membrane was made up of lipid and protein, whereas the cell wall is made up of cellulose, hemicellulose and pectin. There are three main components: cellulose, then hemicellulose, and third is pectin. So plasma membrane has cell membrane has three major components: cellulose, hemicellulose and pectin. Now this cellulose is a complex substance and this gives structural strength to the plant cell. That is why the cells of plant, fungi and bacteria can withstand very dilute external medium. The strength is provided by this cell wall. Otherwise the cell would burst. If you keep a animal cell in a dilute solution, the water will move inside by osmosis and it will burst open. Whereas plant cells do not burst open. Why? Because of this cell wall. That is why the plant cell have greater capacity to survive in a rough environment. They can survive in rough environment.